Hi, Miss Nikki here. Chapter 29, we're in part 1 here. We're going to move through fertilization. We're going to talk about the formation of the zygote. We're going to talk about the process as the zygote is undergoing mitosis and traveling the uterine tube till it becomes a blastocyst and it will actually implant in the endometrial lining of the uterus. On part 2, we'll talk a little bit more about second and third trimesters. We'll talk about labor, talk about milk production and letdown, and then at the end of 29, we'll talk a little bit about genetics. Prenatal period begins with fertilization, so when the secondary oocyte and the sperm unite, and it ends at about 38 weeks with birth. Sometimes they'll break up embryonic into pre and then just embryonic. So pre-embryonic is when the blastocyst actually implants in the endometrial lining. So as the zygote is undergoing cleavage and dividing and moving through the uterine tube, they call it pre-embryonic. As soon as it's implanted, it's now embryonic, and this is about week 3 through 8. So anything after 9 weeks up into birth, that would be fetal period. Most of us could look at this term, embryogenesis, and realize this is the development of the embryo. I like this image. I think it does a good job of showing you kind of what is embryogenesis all of the time until we complete week 8, right? So that's embryogenesis. The pre-embryonic period, where we're taking the zygote and it's being divided and multiplied through stage 2, 4, 8, 16 here. This blastocyst, when it has a very specific trophoblast and embryoblast set of cells, right? Then we're going to see gastrulation. This is when we're going to start seeing the 3 germ layers forming. And this is about week 3. And then for the last 4 weeks or so, we're going to see kind of the process of organogenesis, where we're actually developing or laying down the foundation in the beginning and then further developing the organs. So fertilization, you're going to see the 2 gametes fuse. Hopefully by now I say gametes and you're thinking about egg and sperm or oocyte and sperm, and they're going to form a new diploid cell. Remember that these are haploid, and when we join them together, we now end up with 2 of every chromosome. So it restores the diploid number. The sex of the organism is determined by the sperm. Fertilization is going to initiate cleavage, where we're going to see that rapid division. This occurs in the widest part of the uterine tube, the ampulla of the uterine tube. There is some argument about whether fertilization happens in the infundibulum or the ampulla of the uterine tube. We're just going to stick with ampulla of the uterine tube. Remember, we're talking about fembrae, and then this is the ampulla, and then, this is the infundibulum. Those are the fembrae, and then once it starts curving is the ampulla. So who's to say it's not happening right here? Where is that? Is that ampulla? Is that infundibulum? So no real reason to argue about it. We're just going to stick with ampulla of the uterine tube. Something to notice is that sperm are viable for about a day, about 24 hours, and sperm are viable for several days. So capacitation is to make something capable of. So the sperm have to become capable of fertilizing the secondary oocyte. So remember, we have our flagellum, we have our midpiece, which is going to have mitochondria. Then we have our DNA. Then it's going to have some acrosomal enzymes. And then there are glycoprotein tags. So sugar and protein tags that are hanging off of the membrane here. So in order to release the acrosomal digestive enzymes to get through the zona pellucida, we kind of have to remove some of those proteins and sugars that are on the outside of the membrane of the sperm. And so this is going to last a few hours. You'll read in your book, they talk about calcium. So when the sperm are attracted to the high level of progesterone from the corpus luteum, they know that that's the side that has the secondary oocyte that's just been ejected. We have to pull off some of these sugar tags so that the acrosomal enzyme can do its job to break through that barrier that's on the outside of the secondary oocyte. You can see here the phases of fertilization. They talk about the corona radiata. Remember, these are those granulosa cells that are on the outside. You can see the zona pellucida here. Remember that the secondary oocyte is arrested in metaphase 2. It has not finished meiosis 2. So the sperm are going to reach the secondary oocyte. They're going to push through the layers. They're going to kind of weave through till they're actually in contact with the zona pellucida. And then all of the glycoproteins, or most of them, are off, and we'll have the acrosomal digestive enzymes more easily released to hopefully break down that barrier. So remember, millions of sperm, I think we said 150 to 300 million sperm are deposited during ejaculation. So remember, the vagina, there's an immune system for the female, there's this mucosal layer. It is the lining of the vagina is acidic, so the sperm have to make it from the vagina through all of these environmental conditions, through the cervix, through the uterus, out to the correct uterine tube, to the correct ovary that's just ovulated this secondary oocyte. So they think that it's attracted by chemicals like progesterone. So basically the sperm are kind of smelling which ovary just released a secondary oocyte. Attempted fertilization, when it reaches a secondary oocyte, we only want one sperm to be able to get through into the oocyte. So during this acrosomal reaction, we're going to see those digestive enzymes are going to break down the zona pellucida barrier. It's going to allow the sperm to penetrate the secondary oocyte. And once this reaction happens, it causes the hardening of the zona pellucida. There's also a swelling. So here's the oocyte. The sperm makes its way inside. The DNA gets ejected technically. And we're going to see a swelling of the oocyte due to a calcium channel. And that also hardens the zona pellucida and pushes any other sperm that might be trying to make their way through. So those other sperm are prevented from entering. This ensures that only one sperm fertilizes. So the polyspermy, if multiple sperm, then you're going to have way too many chromosomes, right? You're going to have triplets, trisomies. Remember, we also have to contend with the fact that this oocyte's sister chromatids have not separated yet. And then we have our sperm coming in with 23. Well, if another sperm comes in with 23, that's way too many chromosomes, right? Nice image here. You can see phase 1 where the sperm are navigating the corona radiata. You can see where one sperm, the acrosomal enzymes, are actually digesting the zona pellucida. And then finally, we're going to see the sperm nucleus enter into the secondary oocyte, and that's going to trigger the completion of meiosis 2. So the fusion of sperm and oocyte plasma membranes and the fusion of the sperm and ovum pronuclei. So once they start calling it ovum, they're saying it has finished meiosis 2, right? So secondary meiotic division is finished. And now we're going to take the ovum nuclei pronucleus, right, and the sperm pronucleus, and we're going to fuse them together. So now we have our diploid nucleus, and we would call that a zygote. As the zygote moves through the uterine tube, we're going to see these series of meiotic divisions. We're going to increase the cell number, but not the overall structure. It stays rather small. We go from a 2-stage to a 4 to an 8 to a 16. The 16-cell stage, the morula, is going to continue to divide, and eventually we're going to have this pre-embryo structure called the blastocyst. And the blastocyst has that ring of cells on the outside, which are called trophoblasts, and then it has this inner cell mass, which is called the embryoblast. Nice image here. You can see the development as we move through, and then here's our nice late blastocyst. We can see the trophoblast, we can see the embryoblast. So this is going to become the embryo. The trophoblasts we're going to see are going to become components of the placenta. And here they're just trying to show you that the remaining zona pellucida is breaking down. So it takes us back to our first image where we see fertilization occurring, then we see the formation of the zygote and the zygote as it's traveling down the uterine tube before it gets to implantation. We have to have this structure, this blastocyst structure, for implantation into the endometrial lining. So the blastocyst is going to enter the lumen of the uterus. The zona pellucida has broken down. We're going to see implantation about day 7. It is completely burrowed into the uterine wall by day 9. So we're also going to see a differentiation. So this trophoblast turns into 2 separate layers. One's called cytotrophoblasts and another's called a syncytiotrophoblast. So there's an inner and an outer layer. We're going to see that the syncytiotrophoblast is going to produce HCG. So remember, HCG promotes the corpus luteum. It's positive feedback. Keep doing your job. What do we want corpus luteum to do? Keep secreting progesterone, right? Make the lining of the uterus stay thick. So this is usually what's detected in a pregnancy test. We talked about this a little bit before. Levels are high for the first 3 months. Eventually, the HCG levels are going to drop when the placenta is able to produce its own estrogen to maintain the pregnancy. So corpus luteum is the first chemical that's going to promote increased progesterone, keep the lining of the uterus built up. Once the blastocyst has reached the endometrial lining and it differentiates into these syncytiotrophoblasts, those are going to secrete HCG, which is going to have positive feedback, tell the corpus luteum, continue to release high levels of progesterone and estrogen until the placenta can build up. As soon as the placenta, about 12 weeks, once it's formed, then it is producing the majority of the estrogen, and we'll see a decline in the corpus luteum. Nice image here. You can see exactly how the blastocyst is implanting into the endometrial lining. So you can see blastocyst here, the embryoblast is facing towards the endometrial wall. We can see what was the trophoblast is now differentiating into these syncytio and cytotrophoblasts. And we can see that that syncytiotrophoblast is invading into the endometrial tissue. And what is it really trying to get to? These uterine glands, these pools of mom's blood. Nice image here to show you the high levels of HCG in the beginning. And then we can see that the placenta is going to take over. And then we're going to see a decline in HCG from the syncytiotrophoblast. We'll see a decline in the corpus luteum, and now it is solely the placenta that is producing the majority of the estrogen and progesterone to maintain pregnancy. Every time that we talked about the normal anatomy, we talked about all the things that could go wrong, right? So here, when we talk about infertility, we're talking about the inability to conceive and maintain the pregnancy. So sometimes it's a conception problem, sometimes it's maintaining or holding the pregnancy. And you can see they list multiple causes: pelvic inflammatory, endometriosis, ovulation disorders. This one is kind of new. We didn't talk about it when we talked about the males. Remember that sperm develop after puberty, so it is possible for males to develop antibodies to the glycoproteins that are on the outside of the acrosome that we just talked about. So that would cause your immune system to destroy sperm, and you could have a low sperm count. Remember, males ejaculate hundreds of millions of sperm, but only a few hundred actually make it to the egg and start digesting the zona pellucida. Multiple treatments, right? We can do insemination. We can give you oral medications to cause oocytes to be ovulated. We could do in vitro fertilization. We could also use donor oocytes if something is wrong with your secondary oocytes or you're not able to ovulate. You could use, say, a sister's oocyte. We've talked a lot about chromosomal abnormalities during gametogenesis or gamete development. Remember, in the females, the oocyte arrests in prophase 1 until puberty, and then it rests again in metaphase 2, the second meiosis division, until it's fertilized. And then during fertilization, remember we said that the sister chromatids haven't separated until the male sperm DNA gets deposited. So you could also have a trisomy issue if something happens to this second meiosis division. And then cleavage, as the zygote is copying itself on its journey down the uterine tube, there's lots of cell division there, right? 2 cells to 4 cells to 8 cells to 16. So during that division, you could have some chromosomal abnormalities. They think about 50% of pregnancies are terminated from spontaneous abortion, which we would call miscarriage. So there's some sort of chromosomal abnormality that's triggering kind of this termination. Again, it's if you have too many chromosomes, you have too many proteins being made, and it's just not viable for life. Now we're going to look at the formation of this germ disc. So they use some terms like hypoblast and epiblast. We're going to see that the embryoblast is going to differentiate into layers. So just like we had the trophoblast that differentiated into the cyto and the syncytio, right? So the trophoblast differentiates, well, so does the embryoblast. Not as concerned with these terms. We're going to move past these and talk about the germ layers, but I do have an image if you want to see exactly what we're talking about. So I think it's important to see these structures compared to each other. So here we have our blastocyst as it is differentiating and embedding into the endometrial lining. So we can see this blue mass of cells, the embryoblast, is going to start differentiating into the hypoblast and the epiblast. So the hypoblast is facing the yolk sac, and the epiblast is facing towards the amniotic sac, or what's going to be the amniotic cavity. So then you can also see that the trophoblasts have differentiated into the cytotrophoblasts and syncytiotrophoblasts, and those are responsible for the continuing invading of the blastocyst. So here we are at week 3, and we can see a little bit more development here in this area. And this is when they start talking about these kind of germ layers, this disc, if you will. So that is actually going to become the embryo. So extra embryonic membranes. So these are formed by that disc, and it's going to protect the embryo, assist in nutrition, gas exchange, removal of waste. We have this structure called the yolk sac. It is the first embryonic membrane to develop, and it is an early important site for blood cell and blood vessel formation. The amnion, this membrane is from that epiblast layer. It entirely encloses the embryo in a sac, and that sac is secreting a fluid, amniotic fluid, right? This tissue is going to secrete that fluid and bathe the embryo, prevent it from drying out, and also think about the fact that there are lots of chemical signals being told to this embryo to develop into certain organs. So that fluid makes it easier for messages, chemical messages, to trigger cells to differentiate. You can see here the chorion. This is the outermost membrane. This is formed from the cytotrophoblasts and the syncytiotrophoblasts. It is eventually going to become the placenta. So the site of nutrients exchange between the embryo and the mother. So here we can see we have the epiblast facing towards the amniotic cavity, and that is going to produce that amnion membrane along with the amnion membrane secreting the amniotic fluid. We're going to see the development of the embryo here. And then we have that hypoblast, which was facing towards the yolk sac, and we can see kind of coming off of that yolk sac is this connecting stalk, which is where we're going to see lots of blood vessels. The placenta, highly vascularized structure. So this is the exchange of nutrients and waste and gases. Remember, fetal lung development hasn't happened yet. So we're also transmitting antibodies from mom to growing embryo or fetus. This is also producing the majority of estrogen and progesterone, which keeps the uterine lining built up. So beginning to form about second week, but it's not fully functional and able to take over for the corpus luteum till about 3 months. When we talk about the placenta, most of us can think about what it does, but which portions are derived from fetal or from maternal. So you can see the chorion here is the fetal portion of the placenta, and the maternal portion is that endometrial lining or the pools of mom's blood in that endometrial lining. We're going to see that connecting stalk. Most of us could see this is an early connection to the placenta, and that eventually that connecting stalk is going to form the umbilical cord with the arteries and veins. So part of the placental development is when the chorion develops these chorionic villi. So these are finger-like structures. We have an image in just a second, and these are going to have branches of umbilical vessels. So again, this is gas and nutrient exchange. They want you to realize that mom's blood does not intermix or mingle with the fetal blood. The blood vessels of the fetus are laying in pools of mom's blood. So oxygen is going to diffuse from the maternal blood into the fetal capillaries or blood vessels, and CO2 is going to diffuse out from the fetus into maternal blood. I think this is a nice image here to show you that the chorion develops these chorionic villi. And these chorionic villi have capillaries. And these chorionic villi are laying in pools of maternal blood. So what is the placenta? The placenta is chorionic villi laying in maternal blood supplied by the endometrial, or you could say uterine lining. There are 2 procedures that you can use to check the health of a fetus. They are invasive. Amniocentesis, they would have to take cells from the amniotic fluid, and they're talking about fetal cells. And we could look at the chromosomes, do an analysis, do a karyotype. Chorionic villi, they're actually taking a cell sample from those chorionic villi, blood vessels, and capillaries to look if there's any genetic abnormalities. So this image here just gives you a visual of what they would have to do an amniocentesis. And the same thing with chorionic villi. They'd have to take pieces of the chorionic villi, grow both of them up, add chemicals to them, and look at the actual karyotyping of the genetic information in those cells. They only do these procedures when the risk of something being wrong is greater than the risk of actually doing the test. So usually in older parents or any parents that have a history of genetic abnormalities, would they risk doing these procedures to do karyotype. Now we move to that primary or 3 primary germ layers. So this is called gastrulation. So this is a critical period of development. This is about week 3, and this is where we're going to see the ectoderm, mesoderm, and endoderm. And all body tissues are developed from one of these 3 layers. So this is the embryo structure, but we're actually going to see 3 layers developed from those initial layers that we had. We had a hypoblast, we had the epiblast, right? So we're going to start seeing the development of specific germs. A nice visual here for you. You can see the 3 layers in the embryonic disc. You can see ecto, meso, endo. So we have the ectoderm. Most of us, when we think of ectoderm, we think of the nervous system. Endoderm, we think of all the epithelial linings of all the hollow tubes that we have, right? The digestive tract, urinary tract. You can see several of these glands, pancreas, gallbladder, and then everything else is mesoderm or middle. So this is muscle, right? Skin, all the epithelial linings and blood vessels, lymphatics, serous membranes. So we can see how far we've come. I put this image back in again. Here is the zygote. We've made it to blastocyst. This is when we're implanting into the uterine lining. And then in those 2 weeks up to 3 weeks, we're having development of those germ layers, or that gastrulation. We're going to have this folding process that also occurs in order to develop the spinal cord. And now we're here at about week 4. So now is when we start talking about organogenesis. By about week 8, so we're here, week 8, we have a rudimentary form. So we kind of have this body plan down, right? We have a head, we have a rump, we have limbs. We've kind of framed the house, if you will. We know we've had cells differentiate into different organs. And so we're now moving from 8, which is embryo, into fetal period. So by week 8, we've set the game plan, we're all ready to go, and then all we're doing from week 8 to birth is rapid complex development of these organs that we've already kind of put into motion. So during this time when these organs are developing, so from week 3 here to week 8, this is when the embryo is particularly sensitive to teratogens, or anything that can cause birth defects or death in the fetus. This could be alcohol, tobacco, drugs, some viruses. I think we had the Zika virus for a while there. Pregnant mothers were getting bit by mosquitoes, transferring the Zika virus, and it was causing some developmental issues. I think hydrocephaly. So things that can pass from mother to infant that can cause birth defects. So now we're in that fetal period. We're just seeing the maturation of the tissues and the organs. So we're talking fetal period, 9th week until birth. So rapid growth. But I want to point out, we're still at, when we start the fetal period to when we end the fetal period, this is a lot of growth. Think of this as about the size of a grape or a cherry. So this is a really small structure here when we start week 9 until we have this rapid maturation of tissues and organs till we get to what would be a baby that is birthed at 6-7 pounds, right? So a lot of weight increase during the last 2 months. Pregnancy is divided into trimesters. So the first trimester, first 3 months of pregnancy, this is where we have the zygote, become the embryo, and then become the early fetus. So remember, 3 months is really about 12 weeks, right? Then we're going to see the second trimester, where you're going to see the growth of the fetus, expansion of the maternal tissues, and we'll talk a little bit more about this. And then we'll see the third trimester, where we're going to see the fetus is growing the most rapidly, and then mom's body is also preparing for labor and delivery. So we'll talk more about oxytocin in part 2. So basically, we've covered this first trimester, and we've talked a little bit about the fact that we just have maturation of the fetus tissues and organs. So